Recently, I got the COVID-19 vaccine, and for quite a while, I've been hearing this term mRNA vaccine being passed around. However, what exactly is the difference between this mRNA vaccine and the vaccine that many of us have had since we were children? Well, it turns out in order to understand how mRNA vaccines work, we first need to understand the process of protein synthesis in the body. Now, mRNA, also known as messenger RNA, is a type of ribonucleic acid similar to DNA except for two key differences. Instead of this famous double helix structure that DNA has, RNA consists of only one strand, and instead of the nucleic acid thymine, it contains uracil. Now, messenger RNA is essentially a copy of the instructions in DNA for how to make a specific protein, and it goes to an organelle called the ribosome, which reads this instruction set and creates a corresponding amino acid chain, or protein. Now, viruses exploit this system by injecting their own mRNA into the cell, which the cell then reads and begins creating more viruses, meaning the virus has essentially turned your cell into a virus manufacturing factory. Now, typical vaccines are varying in how they work, but usually they function by injecting a weakened or dead version of the virus into the body, which then gets broken down and ingested by your immune cells in the lymph nodes. Now, from here, antibodies develop, which then attach themselves to the virus in order to neutralize it by not only preventing it from interacting with your own cells, but also marking its location for other immune cells. And from here, B cells develop, which are the ultimate goal of vaccines, as these remember a specific antigen and are able to mass produce antibodies if the same virus is ever re-encountered in the body. And furthermore, they can last for several years or even decades. Now, how does this differ to the mRNA vaccine? Well, mRNA vaccines work by injecting a strand of mRNA for a noticeable antigen on the virus. For the coronavirus vaccine, they decided to focus on that special spike on the virus's surface, which allows it to attach to other cells and inject its own mRNA. Now, from here, the process is relatively the same, except instead of the entire virus being manufactured, it's only that small antigen. So what exactly is the advantage of the mRNA vaccines? Well, for one, because they only require a small portion of the virus's genome, once the virus's genome is mapped, they're a lot faster to be developed and distributed compared to typical vaccines, which can often take 10 to 15 years to develop and have a long, complicated, and time-consuming process involved with procuring safe samples of a weakened virus. Furthermore, because the entire virus isn't injected, they have a lot fewer side effects, as even a weakened version of the virus can do some harm to your body, meaning that mRNA vaccines are a lot more accessible to people with weakened immune systems. Fascinating, isn't it? How the very things viruses use to replicate can be used against them.